All right. Thanks for giving me an opportunity to speak in what has been a really wonderful workshop so far. So I want to kind of give a little bit more of an aspirational talk, which at the end I'm going to hopefully land a plane or land the plane and tying it all together in a lot of things we talked about so far. And I think a lot about imaging and cartography and using images to actually be tools. And what do I mean by that? This is a example I almost give at every talk I give if I've at conferences or at universities, because people are like, "Where's PNNL?" And I go, "Well, it's in Washington State." And if you're from the coast, you really have. I've learned that people that are on the coast don't actually know that everything in the middle of the country exists, unless it's Chicago. Um, I go, we're in Washington, and if you've heard of Washington, you think of Seattle, and we're not like that. We don't have trees. We're in the desert, and you can tell that because this image actually informs us, like, oh, this is where all the greenery is. This is where the trees, and this is where that is. So this is using imaging as a tool, and you can also put multi-layers onto that. So this is more of a topography method that you can see some topography within these images. And here you see that there's some mountains here, and maybe predict that's why we have a desert, is because there's a rain shadow. It also shows us, we can look at individual images of different time frames and tell us how perturbations have changed the landscape. And here's a picture of Richland. You can find on Wikipedia right when it was formed in 1944 when they're building the Hanford site. And this is how we've terraformed this area. So I always joke that if you want to know if we can terraform Mars, this is a pretty good example, a use case. And so this point about historical fiducials, right? We can look at other maps and what we knew at the time and how we classified things. So this is a map I have of Washington State in 1980 or 1894. We have a pretty good indication that it was from 1894 because if you look over here, this is Mount Tahoma. This is why we named our supercomputer Tahoma, not Mount Rainier. And you can see that Benton County doesn't exist. This is Yakima County. This was formed after World War II, or as part of the Hanford site and the whole Rattlesnake Mountains. So a lot of the things I think about is this integrative visual omics. So not only doing the imaging, but using these tools for building platforms that can help with like precision medicine, precision cropping, precision regulating of soil ecosystems, if you're kind of thinking about Monet, et cetera. And that really relies on expert cartography and the ability to get really good data. And so this is a couple other folks, think about this. There's this Katty Broner at University of Indiana, does a lot of visualization, and she's in a lot of these NIH programs that think about how we visualize all this data. And she uses this example of a map from 1736 that always talk about this is where we are, probably in all the molecular cartography or chemical cartography we're doing at this point. This is kind of how we can view the world. And this is where we want to be. We want to be in this Google Earth view, where we've already done a lot of imaging, but we can actually go in and point at little things we want to see and then kind of get a better understanding of why things are where they are. And this, to a point we've been talking about, is multi-scale imaging, right? We're looking at the whole Earth. We can zoom all the way down into Mexico City or into Denver or wherever you want to go and see what's there, see a bunch of things that are happening there and understand maybe how the cities are formed, the mountains around them, et cetera. But to do this, I think what I've learned so far and being in all these consortia and doing these things is that doing this requires really large multi-talented teams, diverse skill sets, et cetera. And it requires obviously spatially resolved measurements, so imaging being one of those. It requires traceable and robust measurements. So this goes back to the having standards, having standard data, making sure we understand what we're seeing. And then, of course, this is what we were talking about, the ability to access data using FAIR principles, being able to have reliable and complete databases, everything that's tractable. And then I think this goes back to the other question I was bringing about what images do you want to use, right? For what kind of modality do you want to use? This can be helpful in anchoring, and I'll give some examples of this, but anchoring our data to different imagings. modalities. And then also, if you've worked in multidisciplinary teams, you've realized that everyone has a name for something and it's often different. It's multiple different languages. So we need to be able to understand common nomenclature and or have translation between those nomenclature. And then the other thing I always like to point out is we don't like to think about this, but scientists are very conservative. They don't like to necessarily go against what might be seen as the ground truth or whatever, right? If you see something, you're probably worried that you're not going to get it published because that goes against the counter belief. And this example that Caddy always points out is there was a long time that every map maker kept saying that there's a giant river that basically, the Columbia River basically cut the continental United States in half. And so that was the only way that you could get to California was through this river. Yet a lot of people knew this and they didn't want to go against the counter-counsel, like the counter belief. So it took, sort of going against that and kind of having courage to be like, well, it's just a mountain range. It's not really a river. But so thinking about that on all these different methods of cartography and building those out, I was trying to think about this from the environmental space. And this is something I look at all the time, this ERA-IQ map. And there's so much data in here. And this is how we can use these maps and these images to really understand what's going on and across scales. And this is, I really like this app and this tool. And unfortunately, I use it too much during the summer because of fire prevalence during the summer and the wildfires, et cetera. So this is a couple days ago. Looking back, you can see where all these wildfires are and where, or at least where there's very bad air quality, right? And you can change in there like the types of particle sizes you want to see and their distributions of that. It also tells you kind of the wind currents and so forth. We're mapping that as well, right? And so this is again last Thursday, I believe. And you can kind of see the air quality if we zoom in, because here we're going through a different scale, again, on visualization. And you can see how bad the air quality was at that time. Well, how do we get all that data, right? Like this goes back to these spatial resolved measurements, having very good sensors. This is a lot of sort of grassroots, this example, grassroots data generation. So these array of sensors, a lot of these people you can just buy a sensor, then put it on this website, and it will map all the air quality from all these different sensors across the state or the country, et cetera, and also take some other open source data. But how this tells you about the data, but why do we maybe have bad air quality? Well, we can also understand where the perturbations are coming from in this case. So here we can see, oh, wow, okay, well, there's a fire here over by Yakima. So this is on the Rim Rock fire. And how do we know that? Because we took another piece of imaging data, and this is a picture of where that fire was, right? And you can kind of get an idea of the size, et cetera. So this gives you an idea of how we can visualize by taking a bunch of images, what might be occurring and why we might actually be seeing worse air quality on the east side of the state because of the way that the wind currents are going. And we can now use it to predict future outcomes, right? So at that time, we had a 73 air quality for the 2.5 micron or smaller particles. And we looked at the weather, we knew the wind, et cetera. And we could predict, okay, Thursday, it's supposed to be the next day, it's supposed to be 64. And because we have so much imaging data, we have so much other modeling data, we're getting pretty good at predicting these things, right? So as long as there's not another fire, we can't necessarily tell you that there's not going to be another perturbation like a grass fire right around the Richland area, but that could cause a change. But if everything stayed the same, okay, this is what we should get. And that will help us also understanding it if another perturbation comes along. And this is something we're also trying to do within EMSL. Oh, I don't know why it's showing these slides. I changed this a little bit. Anyways, so this in this part, and probably Shwarup had chatted about this yesterday within TAP. So now can we also look at this in the microscale? And this example from one of Greg's papers, characterizing the chemical composition of these different types of particles, these large particles, the type of day, the chemical interactions are the physical interactions they had with the sun, et cetera, that cause for different chemical compositions within these different particles. And we're also trying to do this across ecological scales within two other areas within this environmental transformations and interactions overarching IRP. And that's the biogeochemical processes. So this is a lot of the things we've been talking about when it comes to looking at mineral organic matter interactions, pore scale, hydrology, et cetera, as well as we're looking at the rhizosphere process. So the perturbation, in this sense, if you think about the plant and the plant root, what is it doing to the soil microenvironment? So we've seen a lot of recent successes EMSL's had in sort of doing this multimodal, multi-scale imaging and using different modalities. One example was from not too long ago, where we were looking at these different biofilms, and we were able to isolate this one layer of this biofilm and try to understand dynamics of carbon and nitrogen cycling and how those were intercoupled. So here we're just looking at bulk data of we give two different types of media to this isolated biofilm. How does it incorporate the nitrogen and carbon that we've been giving it? So we're just doing C13 labeled carbon dioxide, as well as these N15 labeled nitrate or N15 ammonia. And we basically, once you supplement it with a fixed nitrogen source, we thought we would see a difference in carbon metabolism, but we really didn't. And what we were able to do with nanosims, so we're looking at the bulk data now, and then we're able to go into nanosims and look at each contribution of the heterotrophs versus the autotrophs, how they're consuming the nitrogen, how they're consuming the carbon, et cetera. And then also look at the proteomics data and break down what different metabolic pathways might be changing that are regulating the change in carbon and nitrogen consumption. Malak just gave us a great talk about using different types of mass spec imaging across scales, as well as using spectroscopy, in this case, NMR, to be confident about the kind of molecular data that we're observing in both in our MALDI mass spec imaging, and then relating that also to the nanosims as well. So I'm not going to go into that in too much more detail. Yesterday, Odota kind of gave us a really great talk about how to break down and identify these bioactivity hotspots, do some co-localization analysis, and then try to go at a larger scale, a couple X orders of magnitude larger, to understand the spatial distribution of these different types of organic acids that may be causing some of this some of this dissociation of minerals, et cetera, from these fungal species. So that's some recent successive examples. And where do we want to go? Well, Dushin gave this example of being able to image in rhizoboxes. Well, can we use the same technology that we're using there and do it directly within soil? So that's one idea. And this is sort of a larger scale idea that we have of a visual omics platform. Can we take a sample, a plant root or a piece of soil? Can we then get high lateral resolution metabolomics data, in this case, maybe MALDI, where we're looking at things at the single cell or subcellular resolution? Can we use that to inform where we want to do further omics? So this goes back to some of the segmentation we're talking about. What is informative for us to get more information? So in this case, we have this idea, can we do some microdissection and capture the material that we care about, get some more proteomics data, or alternatively, can we also characterize what these proteins actually look like and help predict what their function is? Because the reality of doing all this omics data on microbial systems and plant systems is it's mostly just unknown. We're good at detecting things, but we're like gene of unknown function, protein of unknown function, metabolite of really unknown structure. Can we use that multi-omics information to start informing what those metabolites might be? If we know what the metabolite is, we can maybe start predicting what the protein that structure is that's doing it, and how the gene might be working. And alternatively, can we go the opposite direction? Can we find proteins that might be interesting and then see how then they cause perturbation through the system across the multicellular system or a microbiome? And I always have this idea of spatial phenomics. And can you go and do multimodal imaging of the same sample? Because even though we're talking about this being destructive, like MALDI being destructive, yes, it does consume material, but actually most of the material that we care about, depending on the order of operation, the sample preparation, is still there. So we can still get information about the transcripts, proteins, and metabolomics data from the same sample. And I'm not going to go too much into this, but we have some evidence that we can do these multi-omics measurements on the same sample. So this leads us to maybe a slight break in a conversation, but this kind of goes to what we talked about earlier. Moss started the whole meeting about is that we had this DEOE-BER-ESS annual meeting, and we had a breakout session where we try to ask folks where they see some of the major challenges for chemical imaging within BER and ESS space. And we've talked about them all here. I guess everyone's kind of thinking in the same space. The main answers about what are the grand challenges for chemical imaging, the main things that people would say in these multimeter polls is that scaling, heterogeneity, time, throughput, or dynamics, those are all the major issues. And then also representative and standard data. And then what integrated multimodal chemical imaging approaches are well suited to address these grand challenges? Basically, microscopy-based ones were brought up as the major points, and then high throughput and time resolved measurements, which in a sense, microscopy can do. And then what challenges and needs for data integration are there, and how can we address sort of these things? And machine learning, interoperability, data access, and software were major points. But again, how do we then scale that across? And then again, this idea of standard and robust data and representative data is a major challenge. So those first three questions we asked at the end of this breakout session were all single word clouds. The last question we asked was this fill in what you think are some of the biggest things related to scaling up data information, and how do we go from scaling up the data information from integrated multimodal chemical imaging approaches from models. So this was a more free-form answer. And from the major points that came across was linking field data and sensors, i.e. flux towers, though I will say that I felt like that answer was a little bit skewed because someone yelled flux towers and then everyone started typing it. And then machine learning and pattern recognition. At data synthesis and to help parameterization. And I think that also comes into this idea of registration. This leads into similar questions that we asked you yesterday about the challenges of integration. You know, what techniques would be useful for integrating imaging data? What computational tools do we need for success? What scaling tools are available and need to be developed? And what data would be most useful for scaling and modeling? And I just want to reiterate this point. We ask these questions. You can answer these questions so many different ways, but I think the task, the point that Moss put out yesterday is the goal of this whole exercise is we want to make microscale chemical information accessible and usable for Earth system model development. So we should be thinking about these questions, specifically in this case, with this goal in mind, because like I said, you can answer this question in so many different ways. And I asked this question yesterday about what signal, when we were asking what tool would be really useful for integrating your imaging data. I really think that depends on what the question is, right? So one of the things we use for mapping mammalian tissue, in this case human tissue, we always use microscopy because we can get a lot of morphology, and we have microscopy that we can then align all our other images together, our mass spec images, as well as afterwards, because clinicians use different just histochemical staining to identify where there's disease, fibrotic tissue, et cetera. But by doing this workflow, we can integrate different morphologies from autofluorescence and also different cell types with our mass spec imaging. But the other thing is immunohistochemistry and doing like immunofluorescence, because what we want, the data we really want to understand is what is the different cellular functions within this case, the kidney. So we have probes that specifically identify unique cell types or cell types we care about. We can then register our optical microscopy data across, and we can extract out the mass spec data at each of those locations and measure different metabolic intensities within each of those things. So this is why I was kind of pointing out there, what is the exact thing we're trying to integrate, or what is the kind of data we're trying to get from our microscopy or our imaging tool. And that should answer the question of what is would be most useful for integrating our imaging data. So what I've heard so far from this meeting is, related to the computational tools we need for success, is there's must-have. There's the ability to do data registration and alignment. We need to have that, right? We need to be able to handle data from different instruments at disparate locations, whether it's Runima doing all the multi and other kind of microscopy data here at EMSL, and then sending it to Jocelyn to do all the X-ray, XRF, et cetera, imaging that is happening over at SSRL. Those are two different imaging systems, but they're also in different locations. So we got to think about that. And then we keep bringing up, there's really a lot of considerations we got to consider about distortion issues, image shearing, methods and assays, aberrations. So whether it's nonlinear pixel sizes, depending on our assay, the probing methods or angles. So like when you're doing SIMS, depending on what SIMS you're using, you might have your beam coming in at a 45-degree angle. That can cause shadowing of your images on the other side because you actually can't get ions to that side if you have topography. The other thing is orientation. Jocelyn brought this up earlier about how you take the images is actually how you get the data whether you're probing directly and how if you're taking analysis from the reflective or transmission mode, what angle is everything coming in at, and then of course data weighting. And then as a tool, we need to consider, are we using the right tool, right? Just because you have a hammer doesn't mean everything is a nail, right? So think a little bit about that. Computing capabilities, we heard a lot about that when it comes to if we have a huge piece of data, can we even open it with the capabilities we have? And then human problems, right? We heard about sample prep before and between sampling. Jocelyn brought this up about how from one assay to the other, there's a washing step that then changes the sample a little bit. We consider data pre-processing because we can trick ourselves into a lot of things if we pre-process our data wrong, right? And then, as I noted, sensitivity across methods. Using orthogonal data streams. This goes back to what Malak just talked about, using NMR, LCMS, et cetera, to then validate what we're seeing in some of the imaging data, because the imaging data may not always give us very accurate, in this case from mass spec side, molecular formula or molecular annotations. And then the next question is, what is noise? And is it real or is it an artifact? And should we be using some of that noise? And then, of course, using fair data practices and then collecting and documenting good metadata with controlled ontologies. This is where it makes it so that we can start doing a lot of this connecting of all our different omics data. And then we heard a lot about the scaling tools are available and what needs to be developed, whether it's ImageJ, which is a very popular tool. And then Sam talked a lot about the microanalysis tool today, the PyMS dTorch, and these sparse mixed scales networks that Peter discussed yesterday, and then some of the EMSL map tools that are coming online. And then what types of data are most useful for scaling models? Well, again, you want to ask, what are you looking for? And I think Maruti's talk really captured a lot of the value of using morphology and elemental data and its application for doing the modeling, and then and some of the Porta network stuff as well. And then we want standards that match real world samples, right? Or what we're actually sampling. But we got to keep in mind, all these questions are sort of addressing this main goal of what are the what we want to make microstructural chemical information accessible and usable for Earth systems model development. Okay, so that sort of comes to the end of my talk, and I'll kind of summarize some of the things I talked about, and then what we've heard also here this last 2 days. Why do we care about integration? Well, the idea is we want to do multiplicative analysis, or at least additive, right? One imaging modality can give us some information, but if we can combine the two, we can get a lot more information and maybe have better interpretation of our data. Imaging is a powerful tool, but images as tools are also powerful and helping us visualize complex processes, as I've talked about when it comes to some of the Air IQ information. And then we have computational tools that can help with multimodal image processing and help us reveal biogeochemical processes across scales already. But as we see, there's a lot of opportunity to continue to improve these tools. And that's why Sam comes and gives talks. And I want to try to answer this question, because the more and more I hear about this and thinking about this, what imaging method might help us reach our goals to make this chemical information accessible and usable for Earth model systems development across scales? And I think the winner is just hyperspectral imaging, because it's already scalable. We're already doing it from satellites down to, you can do spectral microscopy, micro Raman, micro IR, etc., using a microscope, and then you can use it from a satellite. So there's a lot of ways that we can already use that. Depending on what wavelength we're using, we can get a lot of chemical information, and that can help us at least anchor some of the other types of modalities and imaging that we're doing, whether it's electron microscopy or mass spectrometry imaging. I think that's one reason why I think it's poised to be very valuable in solving many of these issues. It can also help us capture dynamic processes, as Tomas brought up, and it can be quantitative. And there is infrastructure, maybe we're not using it, but there is at least ideas of infrastructure that can be adapted for real-time processing of this information. And with that, that's all I have.